Dr. Lucas Albano at North Carolina State University. Lucas is a postdoctoral research scholar in Seema Sheth's Evolutionary Ecology Lab at NC State, and his talk is entitled Adaptation to Shifts in Environment in the Native and Introduced Ranges of a Cosmopolitan Plant. So Lucas.
Perfect. Let me just move some of this Zoom stuff out of the way. Hopefully everyone can hear me okay. Yes, as Lauren said, I'm Lucas. I'm currently a postdoc at North Carolina State University, but I'm going to be talking about, I'm going to be talking about my PhD work today, which is on white clover and shifts in environment over space and time in this cosmopolitan plant. So, of course, we know that species can experience many different environments throughout their range. On the screen now, you can see the range of white clover. And a sort of classic question that we might be interested in is how does the same species thrive in these, highly contrasting environments? And one of the reasons for that is a theme that's one of the many themes that's overlapping between Jill's talk and mine is local adaptation, or this home field advantage. You saw this same figure of crossing reaction norms in Jill's talk, where population 1 in red has an advantage in environment 1, and population 2 in blue has an advantage in environment 2 over the opposing population transplanted into that environment. But what I'm curious about is what happens when these environments shift rapidly. And we can think about that in two different ways. One is that environments can shift rapidly across space. So white clover is a species that's native to Europe, but it's been introduced into North America over the past 400 years. And of course, that introduction comes along with some very stark changes in environment between those two continents. So we can think about how adaptation might contribute to invasion success in these novel environments. And another way we can think about it is environment shifting rapidly over time. So we know that the environment is warming rapidly across the globe, and it's still sort of poorly understood how populations are adapting to these environmental changes, or how local adaptation may or may not be disrupted by this change in environment over time. So, sort of overarching question is, can introduced populations of an invasive species, white clover, rapidly adapt to climatic variation in space and in time? So we can address that question with this sort of large-scale transcontinental common garden experiment. This experiment was the sort of bulk experiment of my thesis, PhD thesis with Mark Johnson at the University of Toronto. And we have a whole bunch of populations of white clovers sampled from across a latitudinal transect in North America, source populations represented by the orange dots there, and a bunch of populations sampled across Europe as well, represented by the pink dots there. And all of those populations were transplanted into 4 common gardens in Mississauga, Ontario, in Lafayette, Louisiana, in Montpellier, France, and in Uppsala in Sweden. But in order to be able to test local adaptation in each of these different environments, we need to have some sort of measure of how different these environments are from each other, the source environments and the common garden environments. And we can do this by calculating what we could call climatic distance. So I extracted bioclimatic variables for each source population and each common garden. These were mean annual temperature, annual precipitation, minimum temperature of the coldest month, maximum temperature of the warmest month, minimum precipitation of the driest month, and maximum precipitation of the wettest month. And I combined all of those variables into a 3-axis principal component analysis, which can be plotted like this. And you can see those same populations from North America identified by the orange dots, the same populations from Europe identified by the pink dots, and each of the 4 common gardens identified in green. And then I can then calculate a weighted Euclidean distance between each source population and each common garden as a sort of measure of climatic distance between those source populations and each common garden location. Now we can look at this type of data on a very similar figure as the crossing reaction norm figure identifying local adaptation that you saw earlier, except now we've replaced the 2 different environments on the x-axis with that measure of climatic distance. We still have fitness on the y-axis. And if local adaptation is occurring, we would expect to see this negative slope. So populations that were transplanted very short climatic distances are likely to have a fitness advantage over populations that were transplanted a very far climatic distance. And the nice advantage of having this continuous variable of climatic distance on the x-axis is that we could also use the slope of this line as sort of representative of the strength of local adaptation or the magnitude of the fitness advantage in that particular environment. So in this case, we could say that common garden A is exhibiting stronger local adaptation or a greater magnitude of fitness advantage for local populations than common garden B. So as part of this overarching question, I'm first going to address climatic variation in space. So if we plot our data on a very similar figure as to what you just saw, you can see that strength of local adaptation does differ across space depending on which common garden we're interested in. So overall, there is a signature of local adaptation, but it is mostly driven by a strong local adaptation in Lafayette and in Montpellier. So the more southern common garden location on each continent compared to the more northern common garden location. So we can use this as sort of a baseline result to say, yes, there are differences in the strength of local adaptation across space. But we want to kind of readjust the way that we're analyzing this data in order to address the question of local adaptation between the 2 ranges. So we can take this exact same data and analyze it slightly differently, where if we group the European gardens together here on the right, and also group all of the source populations by what continent they originated in, so Europe or North America. And we can see that the European populations are exhibiting this strong signature of local adaptation. So there's this reasonably large magnitude of fitness advantage for local adaptation or for local populations compared to populations transplanted from further away. Whereas North American populations are showing this flat line, so no signature of local adaptation. And when we look at the North American gardens, we see a very similar result, but flipped. So here, the North American source populations are showing a strong signature of local adaptation to about the same magnitude as those populations were in Europe. But the European populations are not showing any signature of local adaptation, so they have that same flat line. And what we can conclude from this is that white clover has rapidly adapted to its introduced range in North America within the last 400 years since it was introduced. And on top of that, there is a significant cost to that adaptation. So when those North American populations are transplanted back into Europe, they lose that signature of local adaptation. So to answer this first half of our question, can introduced populations of white clover rapidly adapt to climatic variation in space? We do have evidence that yes, they can. So we'll kind of shift focus now to adaptation to climatic variation over time. So in this case, we're curious if adaptation can keep up with climate change. So if we focus our attention just on the Mississauga Common Garden now, and only on populations that were sourced from North America, so just introduced populations and just the northern Common Garden in the introduced range. And if we overlay this kind of simulated change in mean annual temperature data just as a demonstration, where Mississauga would be considered 0, so no change in mean annual temperature from the site of the Common Garden, and very cold populations we'll say are -10, very warm source populations we say are +10. We can plot a curve like this where peak fitness is at 0, so exactly the location of the Mississauga Common Garden. And this is what we would expect if we were seeing signatures of local adaptation. So if a population has been transplanted a very short, either geographic or climatic distance, it's likely to have a fitness advantage in the Mississauga Common Garden. But there's another scenario, which is an adaptation lag. And in that case, peak fitness will have moved to source populations that are warmer or slightly warmer than that of the Common Garden. So I can kind of flip back and forth here, and you can see this shift in peak fitness towards populations from warmer origins. And that would be indicative of an adaptation lag. And when we plot our actual data, we see some evidence of that actually occurring. So if we have proportion of plants survived on our y-axis, we can see that there's a 3.4 degree shift in peak survival towards populations of warmer origins. And that the shift in that peak is significant from a curve centered with a peak of 0. So in the case of proportion survived, we do see evidence of an adaptation lag. If we're looking at seed set mass, there's a 2 degree shift in that peak fitness, but that peak shift is not significantly different from a curve with a peak centered at 0. And lastly, we can look at maximum plant area, which is important because white clover can also produce vegetatively, not just sexually. So we need this sort of measure of how far the plant is spreading as a kind of proxy for its ability to reproduce vegetatively. And in this case, there's a 2.1 degree shift in peak fitness, and it's sort of marginally significantly different from a peak or a curve with a peak centered at 0. So we kind of have a mix of results across these 3 response variables for the evidence of an adaptation lag due to climate change. So in response to our question about introduced populations of an invasive species adapting to climatic variation in time, I guess the answer in this case would be maybe, and it sort of depends on what response variable we're looking at. So just to wrap up, we found that the strength of local adaptation is variable among environments, and importantly, that led to this result of evidence of rapid local adaptation in less than 400 years post-introduction to North America. And we have some evidence of an adaptation lag in introduced North American white clover populations, but the results there are still a little bit mixed and potentially unclear. So I think we need some more studies on that particular aspect of local adaptation or disruptions of local adaptation. So from this study, we can conclude white clover populations are adapting in space, maybe and maybe not in time. So to speak to further studies on this aspect of local adaptation or disruption of local adaptation, I'll offer a quick plug to my current work, which is with Seema Sheth at North Carolina State University. And we're looking at evolutionary rescue in space and time in Mimulus cardinalis. So we have 6 source populations from the north, center, and south of Mimulus cardinalis' range, and 3 common garden locations, again, north, central, and south, and also seeds harvested from before a prolonged heat and drought event and after a prolonged heat and drought event. So we can look at evolutionary responses that have occurred as a result of that event. If you're interested in that or any of my white clover work, my email is again on the screen here. And I'm also happy to answer any questions that you might have in the chat or in the discussion session later. Quickly, I'd like to thank my co-authors. In particular, I'll shout out Mark Johnson, my PhD supervisor, and Nick Coyers, who was our collaborator in Louisiana and who also has a talk in this virtual symposium. Also like to thank all the people that helped with the actual physical work of this experiment, which was a very large endeavor, and the organizers of the symposium. Thank you very much.
Thanks very much, Lucas. So we do have quite a number of questions that have come up in the chat. So what we're going to do in this chat, in this Q&A and in this question and answer session is we'll first ask Lucas to address some of the questions that are addressed to him, and then we'll also ask Jill to answer some of the additional questions that have come up for her as well. And some of the questions can be addressed to both, of course. So one of the first questions on the chat was a relatively straightforward one, and that is, have you published your work on the common gardens?
Oh, okay. Yeah, I guess that is a straightforward question. Not yet. It is available on bioRxiv right now, and we're kind of working on some very final edits before resubmission. So it's available on bioRxiv, and hopefully the kind of like final polished version with some helpful reviews will be out very soon.
Another question, this one is more of a scientific question, is do you think this adaptation is relatively fixed, or could the adaptation rapidly revert within a few generations?
It's a good question. I guess the actual speed in terms of generation time, we don't really know. So yeah, the best evidence that we have is that there's kind of mixed evidence for whether adaptation can occur within a few generations or not. Of course, 400 generations or 400 years worth of generations is relatively fast in terms of adaptation to the introduced range. But there's also evidence in other species that local adaptation has occurred in introduced ranges in as little as like 20 years or 50 years. So even faster than white clover. So yeah, I guess it's possible that adaptation can occur on a quite a bit faster timescale than we've seen so far in white clover.
Another question concerns the sources of seeds for adaptation. Could some of this adaptation be attributed to selective cultivation, given that Trifolium repens is widely used in agriculture?
I think yes. I think there's a good argument for that, because yeah, it's not just a natural species that's not being moved around intentionally by humans. So that's definitely something to consider. I guess we do have evidence from Nick's work that it is the sorting of existing genotypes that's primarily responsible for, I guess, the recreation of clines across the introduced range, in particular with the hydrogen cyanide polymorphism as the chemical defense polymorphism in white clover. So I think that sort of provides some backing to rapid local adaptation as being definitely a primary factor in the sort of sorting of these genotypes across these large environmental gradients in North America.
So this next question seems to be more of a broader question that's addressed to both that either you or Jill could address. And it's so I'll let either of you take a stab at it. How do you tease out the potential effects of harvesting, seed storage, planting methods, and timing from climate adaptation? This is sort of when you're running a common garden experiment.
I guess the short answer is that I haven't teased out all of those potential effects. We definitely try to consider all of these different things, not necessarily accounting for them all at any particular time, but definitely trying to keep them consistent across common gardens so that they're not affecting things in the exact same way or in different ways, sorry. But yeah, you can only account for so much, I guess, would be the short answer to that question. So I guess trying to maintain as much consistency as possible is at least my approach.
Jill, do you have any comments on that, since it sort of is an issue we all deal with when we run these kinds of experiments?
Yeah, sure. So most of the seeds that I used in the experiments that I showed you today were seeds that I collected about a decade ago, and basically in about 2012 or 2013. And then we run our seeds through a refresher generation, a generation to generate inbred families and to homogenize maternal effects. And so we're treating all of the seeds similarly across all of the gardens. But we've also gone and done collections more recently. So we do these refresher generations relatively frequently when we can, so that we're using fresh seeds in the experiments. But I have collections now I did a ton of collections this year, and we did a bunch of collections last year as well. So ultimately, we're building toward potentially doing a resurrection study, but Bucharest stricta is a perennial. So I don't think 10 years will have been enough time to actually notice any difference, any evolutionary difference between ancestors and descendants. It could just be 1 to 2 generations. So that's going to be a longer term project. I love resurrection studies, so they're a really neat method for addressing this issue.
So this is another sort of broad question that either you or Lucas could address, and that is there a way to determine how long it takes for introduced non-native species to adapt to new locations?
A good question. This is maybe a sort of a cop-out answer, but I don't know if there is like such an endpoint to whether they're adapted to a new location or not. They're presumably very poorly adapted when they arrive to that novel range, and then provided that environment stays constant, they would be expected, if they're successful enough in that novel range, to just continuously adapt to that range. So yeah, I don't know if there is a definitive way to determine how long it's going to take for that adaptation to occur. And of course, it'd be different for every species, depending on, or every introduced population, even depending on the characteristics of that species or standing genetic variation within that population.
Yeah, I find it remarkable that what is it, 150 or more years since Darwin, and we still can't predict how rapidly evolution will take place. Although we can model it, I suppose. But as you say, it's going to be highly variable across different situations with generation times, migration rates, and so on and so forth. So it's a tough one. Jill, do you want to answer to that, to add to that?
I'm not sure that I have a great addition to that. I haven't been looking at invasive species or introduced species for a really long time. So I think Lucas did an excellent job with that question.
Okay, we'll move on to the next question. I think it's a really interesting one as we face with climate change is asking really whether plants can adapt fast enough. In many situations, like in your species, it seems unlikely unless there's lots of migration. And it's a relatively short-lived species, so things will be worse for other stuff. So it's something that's super fascinating. But I'm not the one supposed to be discussing these. So here's another one. This one is for Lucas. Was this species of white clover collected from disturbed sites, undisturbed sites, or a mix?
So I guess white clover in particular is a species that thrives in disturbed sites. So I would say the vast majority of the populations were from sites that would be considered disturbed. I didn't collect the European populations. Those were borrowed from another study. But at least as far as the North American populations go, which I did collect myself, most of them were taken from lawns. So city parks, schoolyards, things like that. What I try to account for when choosing all of the sampling sites is avoiding urban or very large urban areas, because we know that urban-rural gradients have a substantial effect on evolution in white clover. So everywhere that I sampled from was generally a very small town, 5,000 people or less. And yeah, I would just sort of pick whatever kind of type of city park was available that had a reasonably large lawn and walk around and harvest the seeds that way. So I guess, yes, you can consider all of those sites to be disturbed.
So another question for you, Lucas, is how are you administrating the mimulus plants to prolonged heat stress, and what is your control?
So in the case of the common garden experiment, they aren't being exposed to heat stress, at least not directly. And there isn't a control necessarily. We just have populations or seeds from each source population that were harvested from 2010, which was prior to the prolonged heat and drought event, and from 2017, which is after the prolonged heat and drought event. So the comparison there is not necessarily a treatment versus control, but the evolutionary response that we would have seen between those ancestral seeds from 2010 and the descendant seeds from 2017.
There's some pounding out of my window, so I muted myself and then forgot to unmute. So another question to Lucas, and then there'll be one to Jill, and then we'll give everyone a 5-minute break before the session comes back. The question is, how much do you the question is about the microbiota of clover, and how do you think that it may contribute towards plant fitness? And in white clover.
Yes, a lot. So I guess another student in Mark's lab who defended right around the time as I did. Most of his PhD thesis was focused on local adaptation in white clover in terms of its microbiota, and essentially are the microbial communities or the rhizobia communities within white clover locally adapted to the white clover populations. And I can't speak in 100% detail to the work, but the short answer is that across urban-rural gradients, yes, the rhizobia communities and the white clover populations were locally adapted to each other. So if you kind of scale that up to these large latitudinal gradients or across continents, across ranges, definitely there's potential for the microbial communities associated with white clover to contribute to local adaptation. We didn't account for it in my study. The seeds were just planted directly into the natural soil. But yeah, definitely something that could be contributing. And I'll plug that student's name in the chat for you so you can check out their work if you're interested.
Okay, so this is going to be the last question for the morning session. It's for this session. It's directed to Jill. Has there been any changes in the flowering phenology of plants, like early flowering in either the common garden or reciprocal transplantation experiment?
So this is a perfect question to end on because our next speaker is going to be David Inouye. And David has done incredible work on plant phenology, reproductive phenology in populations in the entire plant community around the Rocky Mountain Biological Lab since the early 1970s. So about a decade ago or so, early 2010s, I was giving a talk at Rumble and David came up to me afterwards and said, Jill, I have some Bucharest stricta phenology data. Do you want it? And I said, yes, please. So Bucharest stricta has been advancing its timing of flowering in David's data by about 3.7 days per decade. As of the last time I analyzed those data, David would have more recent numbers, but that number is very similar to advancements in flowering phenology in other species in that system. And in fact, in lots of early flowering species worldwide, they're all advancing in a pretty consistent way. I'm really excited about our flowering time results. And I'm actually looking at a slide from another talk that I gave about flowering time. But we see some pretty severe constraints on shifts in flowering time in our lowest elevation sites. And our two lowest elevation sites, it actually looks like early snowmelt delays flowering, whereas in our higher elevation sites, early snowmelt is advancing flowering to the same extent that we see in David's long-term data set. So that leaves us with questions about whether constraints on the expression of plasticity could potentially be more pronounced under harsh environments and low elevation locations, or whether delayed phenology may actually be advantageous there because it might help those plants get over the period of time when they'd be exposed to frost. So if you can hang on for another 6 to 8 months, I will write that paper. And yeah, that's the next thing on my docket.
So I want to thank both Jill and Lucas for 2 wonderful talks and for answering all of these questions. We're going to take a 2-minute break. Sorry about this. But please come back at 12:15 Eastern time, 9:15 here in Vancouver. And for the next talk, which will be David Inouye, as has been heralded by Jill's comment. So thanks again to both Jill and Lucas.